Okay, class, time for meditation.
Did you hear what Frank had for ground code the other day? I mean, like, we have to pay a lot for his tuition.
Miss
like,
Quinn!
Come on, guys, what Miss Kid doesn't know won't hurt.
Kenneth, be quiet. Class, this actually brings up our lesson for today. Silence. Sometimes the first method doesn't work. But this isn't the only strategy to silence others. See, that worked much better. Similar to us, plants have two different pathways for silencing, but they do it for their genes.
Genes? Like, genes?
No, I think you're not getting it. Alright then, it looks like we're going on a field trip. Everyone, hop onto the magic school bus.
Seatbelts, everyone! Please let this be a normal field trip. With a friend? No way!
Cruising on down Main Street, you're relaxed and feeling good. Next thing that you know, you're seeing octopus in the neighborhood, surfing on a sound wave, swinging through the stars. Take a left at Human Testing, take your second right past Mars on the magic school bus. Navigate a nostril, climb on the magic school bus, spank a plankton too. On our magic school bus, drop the river of lava on the magic school bus. Such a fine thing to do. So strap your bums right to the seat. Come on in and don't be shy. Just to make your day complete. You might get baked into a pie on the magic school bus. Step inside, it's a wilder ride. Come on, ride on the magic school bus.
Where are we going, Miss Quinn?
Isn't it obvious? We're going to a greenhouse. Hold on, we've arrived.
Wow!
Yeah, I know. Arabidopsis is actually used as a model plant system to study higher up organisms. They're pretty awesome, and they silence themselves, just like how we silence each other in class. But they silence their mRNA. Come on, class, let's take a look at what they're doing in the nucleus. So in reality, a lot of mRNA transcripts are made that are not necessary. And Arabidopsis has evolved two separate pathways to deal with this problem. But both pathways begin with the creation of unprocessed miRNAs. miRNA stands for microRNA, and they're tiny non-coding RNA molecules that silence other RNA molecules. Kind of like a teacher in a room full of chatty students.
Oh, and yeah, look here, it's DRB1. Double RNA binding protein 1, or DREB1. It's a transcription factor that enhances the function of DCL1, dicer-like protein 1, which processes the pre-miRNA into miRNA that can be transported out of the nucleus. Isn't that right, Miss Quinn? Double RNA binding protein help dicers and dicer-like proteins bind so that they can process the microRNA?
You got it.
The miRNA then binds to the targeted mRNA that needs to be silenced. AGO1, or Argonaut1, mediates the mRNA-based cleavage splice.
So this pathway physically cuts up mRNA? That's so rad!
Effectively splicing the mRNA.
But what happens when the plant is in a different developmental stage and it can't use the first pathway? Is that really the only option?
Good question. It's actually known that Arabidopsis has a secondary pathway that also silences mRNA, but just in a different way.
However, instead of DRB1, it uses DRB2 as a transcription factor that enhances the activity of DCL.
And instead of just AGO1 acting to splice the mRNA, an unknown AGO with DRB3 and DRB5
Binds to miRNA, or RNA complex, and undergoes translational progression-based silencing. I've heard all about this before in my botany class. The tertiary complex is actually called RNA-induced silencing complex, or RISC.
So basically, it's just a cluster of proteins that work together to silence the mRNA, but doesn't necessarily chop it up, like in the first pathway.
Yeah, pretty much.
Ooh.
I don't get it though. Why do we even care? I mean, who wants to study boring plants?
Oh, Jeff, you have so much to learn. Let's make a little detour before we go home.
Ew!
Whoa.
I know, right? Silencing would be pretty great if it could stop this tumor, wouldn't it?
But isn't DRB5 just in plants?
No, wait, I get it. Even though DRB5 might not specifically exist in humans, the silencing pathways do. The dicer-like and AGO proteins are relatively conserved across the domains, and therefore, if we can find a homolog of the DRB proteins, we can utilize the miRNA silencing system as a form of cancer therapy, right? To stop the tumor from growing.
Oh yeah, I heard of this gene, TRBP, that acts almost as the human version of DRB. So using the plant model, couldn't we implement what we know about DRB silencing into humans?
You got it, class. In fact, if anyone's interested, Professor Slack's lab, the other teacher in elementary school, recently published an article titled Micro-mRNA Silencing for Cancer Therapy Targeted to the Tumor Microenvironment that covers exactly what we just talked about. In fact, let's make that our homework for tonight.
Whatever happened to predictability? The milkman, the paper boy, evening TV. Did I get delivered here? Somebody tell me, please. This old world confusing